Cell division or mitosis is a complex process essential for growth, repair, and development. As you've just seen, imaging allows us to capture information about cellular processes like mitosis in both time and space. However, when we quantify these images, we get hugely complex data containing thousands of features for every cell. Manifold learning allows us to represent this data in a 3D visualizable and more interpretable form. However, it's still often challenging to form hypotheses and draw conclusions from this data alone. This represents a disconnect between our ability to perceive visual information and couple it with the numeric data we derive from that information. To bridge this gap, our lab collaborated with Thomas Bednarz and colleagues from UNSW's EPICentre to develop a visualization software called BioDive. And I'll show you a quick demo of how we can use BioDive to detect and analyze cell division in prostate cancer cells. So here we are in BioDive, and the first thing I'm going to do is load in my data. So this data set contains information from around 80,000 prostate cancer cells that we captured in our microscope images. So here we see our manifold embedding that represents the complex heterogeneity of our data set. Each blue point is a single cell defined quantitatively by 750 unique variables. To help understand the structure of the data, I'm first going to color code those cells as either dividing or non-dividing based on a previously defined machine learning model. I'm next going to switch to a different manifold embedding method. Now this is the exact same data, it's just represented in a different form from a different statistical algorithm. As I mentioned, what's great about BioDive is the link between our image and numeric data. We can select cells from within the manifold and assess how they change both visually and quantitatively throughout different areas of the embedding. For this analysis, I'll start by making a selection of mitotic cells in the upper right region and adding them to a set. I'll then do the same for our interphase cells and add them to a different set. So we know these cell populations differ statistically, given that they appear in separate regions of the generated manifold. But this doesn't really help us detect or comprehend those differences. Instead, I'll bring up the images corresponding to each set of cells. As someone familiar with mitosis, I can discern some obvious differences in appearance between these two cell groups. From that inspection, I'll then add some quantitative analytics by building a parallel coordinates plot and selecting some quantitative features that correspond to those visualizable differences. So now we can see that mitotic cells are quantitatively smaller than interphase cells and do not express as much fibrillary, the red marker, or alpha-tubulin, the green marker. I can also use these sliders to directly filter the images and highlight cells with particular quantitative characteristics. So here biological experts can leverage their visual familiarity of cellular processes to map that understanding into the less familiar statistical space. Now, this is an intuitive process that allows us to understand some really quite complex data in a comprehensive and really rapid way.